Hello, everybody. My name is Dr. Dorina Petrovsky, and I am here to moderate today's webinar. I am an assistant professor at Duke University School of Nursing, and I'm also a member of a biobehavioral rig of the Southern Nursing Research Society, which is presenting or co-presenting today's webinar. The webinar today is titled Accelerating Biobehavioral Nursing Research to Achieve Health Equity Using Secondary Data Analysis. In this webinar, we will present an overview of a study called MURDOC, which is a community registry and biorepository, as well as talk about research priorities and set by the National Institute of Nursing Research. The webinar will end with enough time for question and answer discussion on how nurse scientists can formulate research questions and hypotheses based on secondary data analysis to accelerate biobehavioral nursing research. As the presentation is going on, I welcome your questions in the chat that I will monitor. And then at the end of the presentation, I will forward those questions to our speaker. Today, we have 3 speakers, including a surprise guest who's joining us, and I will introduce each of the speakers right now. Dr. Sharon Dougherty is an associate professor and vice dean for research at Duke University School of Nursing. Dr. Dougherty's research is aimed at improving outcomes for children, adolescents, young adults, and families undergoing treatment for life-limiting and chronic conditions. Dr. Dougherty studies how to improve care models, symptom management, and decision-making from diagnosis through the end of life. She has methodological expertise in the use of qualitative, mixed methods, trajectory science, and visualization methodologies for complex data explorations and intervention development, as well as testing. The second speaker is Dr. Julia Walker. Dr. Julia Walker is the Helene Fuld Health Trust Distinguished Professor of Nursing at Duke University School of Nursing. She also holds the rank of Professor of Medicine at Duke University. She received her PhD in physiology from Queen's University in Kingsland, Canada. Following postdoctoral studies at Duke Department of Cell Biology, she joined the faculty in Duke's Division of Pulmonary Medicine, where she progressed through the ranks before moving to the School of Nursing. Her research focuses on understanding the mechanism by which beta-arrestin proteins regulate G protein-coupled receptor function. These receptors, the largest family of cell surface receptors, are implicated in numerous diseases, and therefore their signaling pathways are prime targets for therapeutic interventions. Dr. Walker also holds the position of director of the Duke University School of Nursing Biomarker Laboratory, and the purpose of the laboratory is to facilitate the inclusion of biomarker data, particularly biomarkers of stress, aging, and disease in nursing research. And the third surprise speaker that we have who joined us today is Dr. Kays Gadumi, who is a co-PI in the Duke University School of Nursing MURDOC project. As an assistant professor and data scientist in the School of Nursing, whose research interests are in the application of machine learning and AI in healthcare. So I wanted to, before we begin, I wanted to set a reminder that the Southern Nursing Research Society has an upcoming conference, 39th annual conference, taking place in Memphis, Tennessee at the end of February. And I also wanted to pass along thank you to this year's conference sponsors, as you see here. So Dr. Dougherty, take it away.
Thank you, Dorina. And also my deep thanks to the SNRS Biobehavioral Special Interest Group for inviting us to talk about this project that we've had ongoing for about 2 years now. It's been in the planning stages for about 2 years now. So I just wanted to give you a bit of the background story on why our School of Nursing became very interested in finding a large data set that included biomarker data in it. And it had to do with our strategic planning process that we underwent starting in 2022. We decided to really refocus our vision for nursing science in the School of Nursing. And so we undertook a really in-depth evaluation of where our faculty were conducting the research and how we could better align our vision of our research with what was going on nationally related to health equity. Next slide, Dorina. So we all committed to a new mission statement at the School of Nursing, and within that mission statement was our commitment to advancing health equity and social justice by preparing nurse leaders and innovators who had a commitment to improving health outcomes through transformative excellence in education, clinical practice, and nursing science. Next slide. And so in our nursing science mission, we then decided that we wanted to really focus on expanding the breadth and depth of our Doosan research portfolio in health equity, social determinants of health, and nurse-led models of care. And this would help us move more towards the first priority, which was to develop a more cohesive school-wide vision for our research, who we wanted to be known as. You know, we were at the developmental stage of our research where we had a lot of really excellent programs of research in chronic illness prevention, all along the trajectory of care. But we wanted to be able to indicate some key areas that we wanted to really focus in terms of the depth of our research that would link it up to health equity, social determinants of health, and nurse-led models of care. Next slide. And importantly, we wanted to align with the NINR strategic plan that had had been just published around the time that we were working on our strategic plan work. And as everyone, I'm sure, on this webinar today is aware, the NINR really refocused their strategic plan to lead nursing research to solve the pressing health challenges and optimize health and advancing health equity by bringing in these research lenses. And so our strategic plan really mirrored that, in that we wanted to encourage faculty to continue in their expertise that they had been developing over the years, but putting new lenses on and asking new questions in their particular fields. Next slide. And also the key research priority areas for NINR really aligned well with what we had wanted to do with our strategic plan. But in particular, one area that we wanted to really build depth on in the School of Nursing was in multi-level approaches, bridging biology to society. We had several faculty who for decades had been working in biomarker data and had really done some exciting things that would allow for hypothesis testing related to where people live and work and spend their lives, how that impacts their biology. And in particular, we had a great leader in Dr. Walker, who was there to help build out the skill set of our faculty, but we wanted to broaden it. We wanted to broaden the depth and breadth of our faculty. And so we decided then that we wanted to look for a large data set that included biomarker data that we could fund some studies of faculty and expand the expertise across our faculty in this area. Next slide. And so Dr. Walker approached me back in 2022 with an opportunity that she had been approached by the Duke Clinical and Translational Science Institute that had been the home for a very large study called the Murdoch study, which stands for Measurement to Understand Reclassification of Disease of Carabas and Canopolis Community Registry and Biorepository. Very large sample, longitudinal cohort recruitment from a 20 zip code region in the southeastern United States. And our team today will give you more details on that. The original funding for the Murdoch study came from the David H. Murdoch Institute for Business and Culture, with some operational support from the Duke CTSA and the Duke CTSI. But they were beginning to run out of room to store all of the samples that they had. And they also wanted to build out opportunity for others at Duke.
I know I'm supposed to be a jail guy.
And so we said we were very interested and we wanted to see how we could partner with them to purchase this data set, and that would allow us to encourage our faculty to work in biobehavioral exploration of how social determinants of health operate in human biology. Next slide. So we'll turn it over to Dr. Walker.
Actually, I think Dr. Kazumi.
Oh, got Dr. Gahumi, sorry, Case.
Yeah, that's fine. Thank you, Sharon, and thank you, Darina, for the invitation. So I'm gonna spend the next couple of minutes, maybe 10 minutes or so, to give you an overview of this Murdoch, the Murdoch-Luson data. So now we call it Murdoch-Luson data sets. It's actually a community registry and biorepository. This is a longitudinal community-based cohort of what we have now is 11,774 participants that they were enrolled between 2009 and 2016. The median years of enrollment is 6 years, and we have about 89 of these participants who are still living with high community engagement for prospective work, which is one of the strengths of this data set, that we can call many of these participants, a big majority, in fact, to prospective work in this study, in this project. So these participants were enrolled in a specific geographic area of North Carolina. It's around Cabarrus County, which encompasses about 20 zip codes. And this community has very little influx and efflux over time, so it's quite stable community. The registry is disease agnostic at enrollment. So as such, it kind of reflects the U.S. Population by demography, like common diseases and many other factors. And it's a longitudinal data that are available from an enrollment questionnaire and annual follow-ups. In fact, in the original data set from Murdoch study, and we have about 95% of the data from that original registry. They have about 65,000 years worth of data, follow-up data, because these are yearly follow-ups. And we have about 88% of the participants with at least 2 time points, with high overall completeness of follow-up. We do have some electronic health records available for some participants, not all participants, including domains such as diagnosis, vitals, procedures, labs, and hospitalization. Yeah, you can say I'm going to spend some time just give it like a brief overview, and then I will go quickly on the rest of the slides. You probably can move to the next slide. Yeah, that map shows basically the concentration of participants around the Cabarrus County, and the intensity of the color shows like the density of populations around those blocks. Next slide. Yeah, so like I said, the data set is a longitudinal data set by so at enrollment, the participants were given a enrollment questionnaire, and their vitals and physical exam data were collected at baseline as well as biospecimen. So multiple aliquots got collected. Now Dr. Walker will talk more about that. So the biospecimen were collected at baseline only, but as I mentioned, many of these participants are willing to, come back, and if you want to withdraw more specimen, I think they're amenable to that. And the biospecimen will include plasma, serum, whole blood DNA, and PAX gene RNA, Buffy codes, and Dr. Walker will give you more details about that. And then the participants were called in every year to basically fill in a web-based self-guided questionnaire. And that's how the database is built over years. And again, the median number of follow-ups is 6, but some participants have 15 or 16 actually years of follow-ups. Next slide. So the survey or the questionnaires, so the enrollment survey or questionnaire has 86 questions around demographics, about the health status, health problems, family, medical history, life habits, and so there's the follow-up survey as well, which has less questions, but also includes hospitalizations and medication lists, because these things change over time. So overall, the database has a collection of 135 data elements or variables about these participants. Next slide. Yeah, this table shows the physical exam and biospecimen data that they got collected at enrollment. So you can see that so we have a lot of information about the number of files, is DNA collected or not, serum number of files, and all these valuable information that we will use for a different type of analysis that we are interested in. And so in total, we have 42 variables about physical exams and bio specimen data. Next slide. So now I'm going to go through some of the aspects and just to kind of give a descriptive, high-level descriptive statistics, just to give you an idea about how the participants are distributed across different demographic variables and some social determinants of health variables. So in terms of the demographics, you can see the median age is about 65, and we have more females than males. And most participants predominantly are between, let's say, 40 and maybe 7 years old. Next slide. Yeah, those pie charts show basically the distribution of gender, ethnicity, race, language, and marital status. You can appreciate that this is some more of, we have 60%, 66% female in this cohort, predominantly non-Hispanic and white and English speaking. Next slide. In terms of diagnosis, the top 2 diagnoses are high cholesterol and high blood pressure. About a little over 35% of these participants, about 9,000 of them have high cholesterol and high blood pressure. But you can see, like, we have many diagnoses and conditions are for people who are interested actually to look at, for example, asthma. So we have like a good size, a good number there, like 1,500. If you want to look at asthma or diabetes or depression, or like we have enough participants to meet those criteria for those kind of studies. Next slide. So this histogram kind of show you the distribution of or the number of participants across the area deprivation index. So the area deprivation index is a mapping tool that is kind of displays the relative socioeconomic conditions of neighborhoods using a measure called the area deprivation index. So it's a tool and an index. What we're showing here is the value of the index. The ADI, the index, has been created from a publicly available data. In fact, it uses the American Community Survey 5-year data. So it's an estimate over the last 5 years. So across theoretical domains of income, education, employment, and housing quality, right? And so rankings of neighborhoods are created by comparing the ADI of a neighborhood to both the nation and the state the neighborhood is located within, right? So it's a relative ranking compared to other counties in the states or compared to the nation. And it's kind of a measurement of a deprivation level of a neighborhood, right? As you can appreciate, so we have about 500 participants that they live in a highly deprived areas. I would say most participants live in a moderate, deprivation level. And a good number, 2,000, well, or 2,700 is living in less or low deprivation level. Next slide. Yeah, those are pie charts showing the education and income. I'm just kind of, stop for like a few seconds to let you take a look about how the distribution looks like in terms of household income and education. Next slide. Housing. So most of the participants, 82% of them live in a single family home. Most of them, they pay mortgage. Next slide. This graph shows the pie charts of emotional distress. About 40%, almost half of them, had never experienced any anxiety feeling, and never had depression. And just to mention that these data are pulled up from the survey. So these are the surveys that and this applies to all the pie charts here. So these are coming from the participants. So it's not a diagnosis or anything. It's coming from the survey. So it's a patient survey data. But this is going to be the last slide, and it's showing smoking versus smoking and smoking exposure. Most of them are non-smoking. Half of them said, look, they don't have smoking exposure at work or at home. And I believe this is the last slide, but I will I will say that because we have access to the geospatial or geographic location data of these participants. So what we have is we have the FIPS code. So we have the information about the census block group level, and then the zip codes of these participants. Because we have the access to these type of data, we can actually augment and append the data sets we have with publicly available data sets about all kinds of social determinants of health. And I'm naming few data sets like the Justice 40, for example. The Justice 40 data set has like over 30 variables about social determinants about pollution, access to food dessert situations and stuff like that. So this data set is could be actually made way larger, wider, thanks to access to the FIPS code, which will give us access to a lot of data sets. We can use the SVI, the CDC Social Vulnerability Index, which provides lots of data about social determinants of health. So this is a rich database, could be made richer thanks to access to geolocation data. I'll stop here and now hand it over to Julia.
Thanks so much. I'm going to make a quick question. Do you know if participants are mainly in rural or urban areas?
I gotta rush. I'm in the middle of a class.
I'm sorry?
I'm sorry, let me repeat that. Do you know if the participants are mainly in the rural or urban areas?
The RUCA, I think, data does not define this area as rural, so it's not predominantly rural, no.
Mm-hmm. Thank you, Dr. Walker.
Thank you. Can you see me?
Yes.
Or you don't really need to see me, you just need to hear me. So I'm going to talk a little bit about biological embedding of these social determinants of health. So if you can advance to the next slide, please. So many of you may know this already, but for those of you who are not that familiar with biological embedding, I'm going to spend a few minutes talking about that. So it's well known that social factors and social determinants of health are indeed biologically embedded. And so you can see in the first box there, the middle box, chronic stress, environmental exposure, and major life events. So chronic stress, day-to-day types of stressors, environmental exposure, we're talking about air quality, temperature, chemicals, etc. And major life events, examples would be things like trauma, either emotional or physical or both. And these types of events can biologically embed either non-genetically, which is shown on the left side of the slide, or genetically, which is shown on the right side of the slide. And I'll just go through each of these in the next couple of slides, but you'll see in the non-genetic, we'll talk about HbA axis inflammation, autonomic nervous system, et cetera. So if you can just advance to the next slide, please. Thank you. So this is sort of an overview diagram. I don't have my pointer, but if you look right in the center of the diagram, that's showing the hypothalamus and the pituitary gland. And that is part of the, and then to the right, we're seeing innervation and circulation to, thank you, to the adrenal cortex. So this is a diagram of the hypothalamic pituitary adrenal axis, which of course gets activated anytime we are exposed to a stressor, and that causes the release of cortisol, which is our main glucocorticoid involved in glucose metabolism as well as immune regulation. And then the other classic one is on the left-hand side there. What we're showing are neurons going to the spinal cord. Yes, thank you. And then sympathetic nervous system, preganglionics and postganglionics, actually the preganglionic going to the adrenal medulla, which is causing the release of catecholamines. And what's not shown on here, of course, is that also we have many, many outputs coming from the spinal cord that are innervating several organs. A classic example is innervation of the sinoatrial node of the heart to make your heart rate go up. And we've all experienced this. Somebody can sneak up behind you or some unexpected event, and you can immediately feel your heart rate rise. So the catecholamines then they get released, and the sympathetic nervous system activation causes just a wide variety of cellular and tissue responses. And those are classic. We've been hearing about those for years. A little bit more recent is neuroimmune modulation. So there is actually innervation between, and there's afferent and efferent communication from the nervous system to the brain, and from the brain to the neurosystem. And so if you think about these systems that get activated on an acute and rapid basis, that is highly normal and highly protective for our body. This helps us to achieve homeostasis. But when these systems are activated chronically, then it becomes quite pathological. And what you end up seeing are these classic stress-related diseases. So it makes sense, at least to me, that some of the stress-related diseases are metabolic because we have this release of these glucocorticoids. Stress-related diseases are also cardiovascular disease, which the sympathetic nervous system controls. We have autoimmune diseases that are stress-related. We have neurodegenerative, which has a huge immune component as well. And so these are the typical stress-related diseases and health outcomes from this type of biological embedding. And there are a number of outcome measures that you can make. For example, a cortisol awakening response to get an assessment of the biological embedding non-genetically. Another classic one is looking at C-reactive protein in the plasma, or your lipid profiles. So those we're fairly familiar with. If you can go to the next slide, I'm going to talk about the genetic biological embedding. And this has a name. It's called social epigenetics. And I want to clarify that we're not talking about changes to the genetic sequence. So we're not talking about social genomics. We're talking about social epigenetics, which is basically a modification of the genes that exist and that affects the transcription of those genes. And so it's something that's fairly dynamic. And so you'll see social experience and factors can indeed influence the social epigenetics. And I've got DNA methylation in big bold there because that's the focus of what we're doing with the Murdoch study. But there are other epigenetic modifications to the genes shown there in red: histone acetylation, RNA silencing, and protein folding. And as I said, these modifications can affect gene transcription, which of course can affect health outcomes and or disease. And so next slide, please. So the reason we focused on DNA methylation as a way to examine embeddedness of social determinants of health factors is that Raffington et al. Published this this paper and this very nice diagram in just a couple of years ago in 2022, showing there are a large number of SDOH factors that do indeed affect DNA methylation. Next slide, please. So for those of you who are not familiar with DNA methylation, this is just a quick slide to sort of go over that. So DNA methylation, it examines how CpG sites are methylated. So what's a CpG site? A CpG site in your genetic sequence is a cytosine nucleotide followed by a guanine nucleotide with a phosphate linker in between. So that's a CpG. Now, there are 28 million of these CpG sites in the human genome, and obviously not all of those are probed in these types of analysis. But what is known is that there are regions in the genome of high density of these CpG sites, and they're called CpG islands. And these CpG islands are located very near to gene promoter regions. And so if you can methylate these CpG sites, then because it's in a promoter region of the gene, it can have a greater impact on the transcription. And if you look at the diagram above, you'll see that there's a normal line and then a cancer line. And the line is supposed to be just the genetic sequence, and a black oval there represents a CpG island. So there are multiple CpG sites there. And black represents methylated, and the white ovals represent unmethylated. And so the modification then is whether these CpG islands become hypermethylated or hypomethylated. And generally, this is not always the case, but generally, the more methylated a CpG site is, the less likely there will be for transcription. So it's basically this chemical group basically sterically gets in the way of the transcription machinery and prevents transcription. And so the way these analyses go, the way these analyses go is to have a normal versus cancer, or a control versus whatever your topic of interest is. And you look at these things called DMRs. These are differentially methylated regions. So the output from these types of analyses are looking at, okay, which genes are most highly differentially regulated between your control group and your group of interest. And that would give a clue as to where the genes that matter for the phenotype you're looking at, where they actually are by looking at these DMRs. And I'll show you an output plot of that called a volcano plot in a minute. So, so identification of the genes this way that may be involved in a disease is a mechanism currently being used for disease diagnosis as well as treatment. So this can lead to and facilitate personalized medicine. But for our purposes, for the Murdoch study, we would want to associate these differentially methylated regions with SDOH factors. And our interest is to look at, although we can move to more and more detailed examination, our thought was to look at aging, first of all, aging and the pace at which a person ages, or the discrepancy between your biological age and your epigenetic age, can be a clue as to the health and well-being of a particular population. And that can be related to SDOH factors, and that's already been shown. I'll show you some examples of those studies. Okay, so this is just a brief overview of the DNA methylation. So on the left, you get the blood or a dot blood spot. You extract the DNA. You buy one of these epic array methylation chips from Illumina or other companies. You run them in a very expensive machine, and you get an output basically shown at the bottom on the left, which is a volcano plot. And on the right, there's a bigger version of this volcano plot. And so what this particular study showed was that there were 194 sites. So they had basically an atherosclerotic arteries compared to arteries resistant to atherosclerosis. And what they showed was there were 194 sites, CPG sites, that were hypermethylated, and 164 sites that were hypomethylated. So those showed differences between these two different types of blood vessels. So in total, then, there were 358 differentially methylated regions, or DMRs. And then those data can be used to, when you look at, okay, where are these CPG sites, they came up with, and which ones were the greatest magnitude of differentially methylated. They came up in this study with 18 genes that they now are further investigating that may be involved in atherosclerosis. Next slide, please. So I mentioned that we're interested in looking at an epigenetic clock, just as an overall, I guess, a harbinger or a proxy for health. And so there are several clocks out there. Essentially, these clocks are, people have done these studies to get a DNA methylation profile, much like I talked about before. There'll be certain number of genes differentially methylated, and then these clocks will give you a sense of the epigenetic age versus the biological age, and that will give a sense, if you do linear regression, you can find out if the person is aging more rapidly or less rapidly. And then there are also pace of aging clocks that the epigenetic profile, the methylation profile from that type of clock is one of accelerated aging. Next slide, please. So here are some examples of studies that have been published. I just pulled out a few that are more recent. So this one was published in 2020. And you can see from the title that it's looking at neighborhood deprivation. So this is very much like the ADI, that the deprivation index, area deprivation index, that Case was talking about. And so you can see, I'll just read the conclusion, but basically they found that those living in areas with the greatest compared to the least neighborhood deprivation had higher epigenetic age acceleration. And it was estimated by these 4 different clocks. So as I mentioned, there's several clocks: the Hanham-Fino age, Grimm age, Horvath. And some of these clocks are better for estimating acceleration of aging than others. And that's why it's important to look at a few of them. And I might add that the DNA methylation analysis we're planning to do in the Murdoch study is what we call epigenome-wide. Now, I mentioned there are 28 million epigenome or CPG sites. The array we'll be using from Illumina contains 935,000 CPG sites. But again, the CPG sites that this array will probe are ones that are in concentrated in promoter regions of genes that have been shown to be very important in various diseases and aging, et cetera. Okay, so next slide, please. Here's another example. This is a 2021 paper from Raffington, and basically the conclusion here was if you grow up in a socioeconomic disadvantaged area, then the biological process of aging is accelerated as an adult, and that is further accelerated associated with Latinx ethnicity. And so, what the relevance of doing these types of analysis, looking at social determinants of health factors related to aging, is thought right there. DNA methylation pace of aging might be useful as a surrogate endpoint in evaluation of programs and policies to address the childhood social determinants of lifelong health disparity. So this is where I see, hopefully, our Murdoch studies going, is really revealing what are the factors at a much more detailed and granular level that are most important for aging in the various groups that we'll examine. Next slide, please. And this is just showing you some other types of output. This is from the Raffington paper. So you can see on the y-axis, we've got the Dunedin pace of aging measurement. And it's the one I told you that is designed to look at pace of aging. And you can see that on the x-axis, we have the level of disadvantage. So as the numbers go higher, those individuals are living with more disadvantage. And you can see that there is an association there between neighborhood level disadvantage as well as family level disadvantage. And this is pretty typical to group a number of factors into groupings. I don't think, I would be very surprised if we find one social determinant of health that is the one that is having the biggest impact, just like you never find one cytokine or one lipoprotein. It's a combination of things. Can you go to the next slide, please? And this is just another type of output here, a box plot. And this was just showing on the left that yes, pace of aging is more rapid in the Latinx population. And if you control for the social terms of health factors, then there's no difference. So suggesting that the social terms of health have a impact on Latinx more than the other races and ethnicities. Okay, yeah. And this was the last paper that I chose to present here. This is 2024, so this is pretty recent. And they chose to look at adults who were survivors of childhood cancer. And you can read the question there. Does race or ethnicity accelerate the aging? If so, is that associated with SDOH? And if so, which factors? And this is really sort of getting at the question. They're looking at a specific population. And then next slide, please. I think has the conclusion there. Yeah, so they found that yes, EAA is epigenetic age acceleration. Yeah, they were significantly associated with race and ethnicity. It was higher in non-Hispanic Black. And it was partially mediated by educational attainment and ADI, the Area Deprivation Index. So again, the relevance of these types of studies is to be able to mitigate this socioeconomic disadvantage by determining what social support systems would be most helpful. Next slide, I think, yeah, so this basically just describes the Murdoch study. I've mentioned it quite a bit already, but we've decided to do methylation analysis on 600 individuals from the Murdoch study. And again, since Case mentioned, we'll have geolocation data, we'll be able to link these participants to census tract data, which will give us very, very granular and detailed information about social determinants of health factors. Next slide. I think this is, oh yeah, so this is the methylation array. I already mentioned we're going to use this probe with 935,000 CPG sites. And yeah, so it is an epigenome-wide analysis. So even if our investigators are not interested in clocks, they'll be able to look at the genetic signature or the genetic profile from anything that they might want to. Not everything, but anything that's currently known. That's how these or genes of interest for what's known currently. That's how these chips or these bead chips were designed. And I think that's it. Next slide. Yeah, questions.
Great. Thank you.
You're welcome.
Thank you so much, Dr. Walker. There were a couple of questions in the chat, but we had individuals who addressed the questions, but I think let's see if we can add anything to that. The first question was, can epigenetic alterations be passed down to future offspring? And someone answered that, yes, this is the case. Dr. Walker, anything to add there or do you agree?
Sorry, just was on mute there. Yeah, for sure. Yeah, they can be passed down. Mm-hmm.
And Dr. Doherty, there was a question about collecting other samples for microbiome analysis, and Dr. Doherty answered that question. Chat. There was another question that I received. Is how were the 600 participants chosen for the EWAS?
That's a great question. They have not been chosen yet, and that is the most important part of this entire process in my mind is making sure that we select the right participants to answer the questions we're interested in, and as many questions as others might be interested as well.
Just gonna stop sharing so we can see each other. Someone Stacey commented, thank you. This is fantastic. I'm very interested in adding epigenetics as a component to my stress biomarker research among nurses in the future.
Yeah,
and-
I would just, in response to that, say that I think that's a fantastic idea. This ability to do these types of analyses, so the cost of the bead chips is coming down, the ability to process the data and to analyze the data. I mean, it's moving forward at lightning speed. So I think that it's generated quite a bit of excitement at NIH funding sources, et cetera. So I think it's going to be something that'll be around for a long, long time and will rapidly progress.
Yeah, and Dorina, can I just add to the question about how has the 600 been chosen, or how did we choose the 600? And Julie said that's a really important stage that we're at right now, because we want to ensure that the 600 that we're going to pay to have the methylation analysis done ahead of time will be that sample that a larger number of faculty will be able to use. We really hope, and this goes to the microbiome question, we really hope that faculty will be able to do these analyses and use them as preliminary data for external funding applications, perhaps at NIH. And so we want to make sure that the largest number of faculty might be able to use the data that is the sample, sorry, that is undergoes the methylation analysis.
And a related question before we get to Eric is, are there future plans to go back to the 600 participants to determine change over time with EWAS?
Yes, definitely. That's the promise, is that we have one sample that was taken on enrollment, and we would like to do longitudinal, looking for change over time. And so, but we need additional funding to be able to support that. And so we're really trying to get some big papers, some analyses that Julie will help lead, and then get some papers out that will allow faculty to apply for funding to do the additional data collection.
Great. Eric, you have a question?
Yeah, thank you. It's a fantastic presentation today. I'm sorry that I had to step away briefly, and so I wanted to ask about the data that's available in this data set related to the non-genetic component. So I know you were talking about, I had to step away right after you were talking about ANS and some of those functions. And I know, like in the biobehavioral lab here at UNC, we've done some sociophysiologic data collection looking at like synchronous heart rate variability between like mother and child and those sorts of things. So I was curious what those, if you had mentioned those, I'm sorry, but if you could catch me up on that a little bit in terms of, you've certainly got the genetic data, and it sounds like you're going to be collecting microbiome data. I was curious if there were any of the sort of non-genetic data sources for this cohort.
Yeah, so thank you for the question, Eric, and also thank you for answering the question in the chat.
Oh yeah, sure.
And also I'll say I just toured your UNC biobehavioral lab with Matt a couple weeks ago. Yeah, very, very impressive what you all are able to do over there. So yeah, all we have right now are, typical biometrics like, blood pressure, weight, BMI, that sort of thing. That was all collected on the individuals.
Yeah.
That's not collected on an annual basis, as I'm concerned. But we do have plasma on them, but that was only from enrollment.
Yeah.
Again, the value in that, I think, is longitudinal types of studies. So no, we don't have yeah, I was over there looking at heart rate variability, which is very interesting to me.
Yeah, for sure.
And now there's more and more devices to be able to capture those data, right? So, but I hope that answers your question. That's what we have in terms of non-genetic.
Thank you. And if I could ask just a follow up on some of the anthropometric data that was captured, do you have any measures of body composition in the data set, or is it mostly just heights and weights?
I don't think so. I saw
Okay.
your little capsule over there.
I will add though, Julia, that we do have access to the EHR data that is linked to this
Yeah.
these individuals. So
That's great.
some of that data will be available in their electronic health record.
Yeah, some of them might have, yeah.
Thank you.
Thank you. Yes, thank you, Eric. The question was, can you share, reiterate maybe the big aim of the whole project? I'm not sure if it's the larger database or just the subset that the Doosan acquired.
Yeah, I think I'll start and I'll let Julia add in, but I think our big aim, overarching aim, was to be able to find a mechanism for our faculty to ask questions about health disparities, social determinants of health at the biological level, to look to see for associations of how health inequities, social determinants of health get embedded into biology. And so we thought one way to get this started was to purchase a large data set that had biological data that those that were already doing biobehavioral data could have fun and play with and have access to on their own, but that we could also use as a training ground for other faculty that were interested in getting started but didn't want to lead, but maybe could be part of a larger team. And so we hope to have sort of both pathways into this data set, faculty that will join a couple of teams that will ask some sort of large hypothesis generating questions and get a couple of papers out, and then other faculty that will use it for their own programs of research.
Yeah, the only thing I would add to that is, although it's predominantly white, the participants, there is a really large number of different races and ethnicities. So that is in keeping with our mission for social or health equity. And also another attractive feature of the data set was that some of these, these people have been in this study for years and they are recruitable, and that is something that that the Murdoch team will easily do. So they have access to these people and facilities to recruit them back for any type of biospecimen or information we might want to get. So I think that's a strength as well.
Great. Related to that question, any case maybe you can address that variables that are in a data set about race and ethnicity? Maybe.
Yeah, we do. Yeah, we do have access to the race and ethnicity, and I showed a pie chart about that. So this is a predominantly white population. I believe the number of blacks is about 1,500. So we have 1,500 blacks. Yeah, if you're interested, we can actually bring back the slide that shows the pie chart of gender, race, ethnicity.
That's good. I will do that while we ask the next question. I'm not sure if you have the answer to this question, but someone asked about the availability of the data set. Sharon or
Yeah, so we're really just in the initial stages of making it available to our faculty within the School of Nursing. And so for the next year or so, we're going to be working on that. But as is common practice, we are going to be likely moving into making it available to those outside of the School of Nursing. The whole point of having a rich data set like this is that others can use it. And so again, we're going to get our internal processes set. It was a very expensive data set and in particular the methylation analysis that we're embarking on. So we want to have our faculty have access to that first, and then we're going to set up our processes and procedures to put it in a way that others can access it.
great. I think we addressed all of the questions in the chat. If anybody has any additional questions, I'm just resharing the slide.
Yeah, and feel free if you have questions about the slide.
I actually had a question that I prepared in terms of maybe it's a question for Dr. Walker or Dr. Doherty. What kind of expertise should neuroscientists have on their teams to answer biobehavioral research questions, such as the ones that you presented in this webinar.
Yeah, I'm going to let Julia take that away.
Yeah, I mean, I think if it's probably your first time doing it, then I would have obviously a statistician who's like a biostatistician, someone who's comfortable with analysis of the epigenetic data, possibly you know, a biomolecular biologist, but really, you know, the way this is moving, for example, is to promote usage by any type of researcher, right? And have available data analysis algorithms on the web. So there are companies who are producing this and making them accessible. So I would recommend having some collaborators for the first time or two. But the way this is moving is that you should have access to the tools that you need to do it on your own, and then just the dollars to get the analysis done. But I do believe that that's going to come down in price as well. Hope that answers your
Yeah,
question.
and I'll just add in there that, you know, if you're thinking about doing this more from a unit wide, encouraging your school to purchase this kind of a data set, I think ensuring that your team has, you know, just looking at the team that we have with Julia as a physiologist who has a lot of experience with this type of analysis, but also she has a lot of colleagues that she works with, geneticists and others that we can easily pull in. And then Case, who has the large data set, AI, data analytics, and data management expertise. With this size of a data set, we had to ensure that we had the data management expertise in our Center for Nursing Research to organize this data in a way that faculty could easily access it.
Yes, I meant to add that a data scientist like Case. Yeah, that's very important.
Yeah, and I'll add to that. So there's a specialty, it's bioinformatics. If you are into analyzing omics data, proteomics, whatever, genomics, there is a whole field, it's called bioinformatics, and those guys actually have access to a plethora of tools to analyze. I mean, we data scientists, it's gonna take us some time, right, to learn how to analyze this specific type of data set because it's quite complex and it has there are specific questions we can ask and specific ways for analyzing these data. We can do it, I can do it, but there are people who are specialized in this just type of data. So bioinformatics, access to somebody with expertise in bioinformatics will help.
Eric, anything quick to add before we enter?
Yeah, just really quickly, I hope that the presentation will get people excited about the possibilities of biobehavioral research, and then thinking about, you know, who you need on your team is going to be driven by the question, obviously, but I'm hoping that the curiosity about what you can do and the potentials you can ask, and to say, like, what kinds of questions could I ask of these types of data. So that's my parting gift, I guess, or statement at least, is to say I hope this has been exciting for you. It has been for me. So thank you.
You stole my lines, Eric. I just wanted to thank all of our presenters for this wonderful webinar. The recording of the webinar will be available on the SNRS website. And as a reminder, up on the PowerPoint is the annual conference. It's coming up in February. So I hope to see many of you there. And thank you all. Enjoy the rest of your Thursday.
Thank you.
Bye.
It was great. Appreciate it.
Thank you.